All right, so just some basic stuff to start with. Differences between prokaryotic and eukaryotic. We know prokaryotic means before the nucleus. These are only regular eubacteria and also archaea. They don't have any membrane-bound nucleus. They don't have membrane-bound organelles. But some things they have DNA. Usually it's just one looped chromosome, one circular chromosome. They do have ribosomes because they have to be able to make proteins. Sometimes they have some little extra pieces of DNA. What are those called? Plasmids. And those are important because what can they do with those plasmids? Swap them during conjugation. It's kind of like sexual reproduction. They do have a phospholipid bilayer, pretty much just like ours. They have a cell wall, especially the eubacteria cell wall is made out of a material called peptidoglycan. Now, in terms of us, eukaryotes, and remember, everything on Earth except bacteria and archaea are eukaryotes. We have a true nucleus, meaning it's enclosed by a membrane. Our chromosomes, there's usually a lot of them. In humans, there's 46. And they're not looped. We say they're linear. They're not really straight lines, but they're linear. Phospholipid bilayer. We have all kinds of organelles, membrane-bound organelles. And the advantage of being multicellular, like us, is it allows our cells to become specialized, to become more efficient. And also it allows us to divide up the work. So we become a more efficient organism because of all these different cells that we have. Also remember that eukaryotic cells tend to be way larger than prokaryotic ones. All right, the whole deal for why cells have to be small. Surface area to volume ratio. What kind of ratio do they need? A high. They need a lot of surface area compared to less volume. When a cell gets bigger, what happens to that ratio? It gets smaller. And it becomes harder and harder for diffusion to get things to where? To the middle. So cells need a high surface area to volume ratio. Now if we were talking about a cube, let's say we took a cell, instead of drawing it all round like it usually is, let's draw it as a cube. How do you find the surface area of a cube?
Side times side times 6.
Yep, length times width, or side times side. That'll be the area of each side. There's 6 sides in the cube, so side times 6. What kind of unit are you going to get if you're finding surface area? Something what? Squared. Now volume, to find that, how are you going to find that in a cube? Length times width times height. And what kind of unit are you going to get there? Something cubed. All right, remember our plasma membrane is made out of 2 layers of phospholipids. The unique important thing about them is they have one side, the fatty acid tails that are what? Well, but this side is not polar, and they have another side, the phosphate side, that is polar. Because they have a polar end and a non-polar end, we say they are amphipathic. And what that causes them to do is when we put them in the water, they form a bilayer. Heads toward the water, inside and out, tails away from the water. But really, our cell membranes look more like this, with mostly phospholipid bilayer, but all that blue stuff up there is what? Proteins. Embedded proteins. Things like channel proteins, receptor proteins, glycoproteins. They actually give function to the membrane. And the stuff in bright yellow, that's what? Cholesterol, which makes the membrane a little thicker, especially in animal cells since we don't have what? A wall. Talk a little bit about organelles. The nucleus is sometimes considered an organelle. It's enclosed in a double membrane. It's got pores in it. And those pores basically allow travel between what and what? Yeah, but between what 2 areas? The nucleus and the cytoplasm, right? It's the doorway between those 2 places. And the things that need to be able to move through that doorway are what? The ribosomes. Where are the ribosomes made at? The nucleolus, the little dark spot in the nucleus, the nucleolus. And also the mRNA needs to leave the nucleus and go to where? It needs to go to the ribosomes, where it can be used to make proteins. So those nuclear pores are important. In terms of our DNA, usually, do you have chromosomes in your cells? Nope. Usually your DNA is all unwound. And when it's in that unwound form, it's called chromatin. That's when it's working. That's when it's being transcribed and translated and all those good things. It only winds up when? Yeah, before, during, and right after cell division, mitosis or meiosis. Then it's in the form of a chromosome. And at least in eukaryotes, the DNA is wound around what proteins? Histones. And that lets it organize so that it's easier to make sure that the DNA is split exactly in half. It would be a problem if it wasn't. Just make sure everybody's on. Is everybody on the list? Pass it around to people who aren't on there yet. All right, some organelles we need to make sure we know functions of. Vesicles or vacuoles. They're basically just sacs of what? Phospholipid sacs. They might store water. They might store food. They can store waste. They're storage containers. Think of them like the Ziploc bags of the cell. They hold stuff. The rough ER. It's usually connected up to the nucleus. The ER, first of all, is like a system of tunnels. We said to think of it like what part of a factory? The conveyor belt that moves things in the cell from one place to another. It's covered with ribosomes. Its job basically is to move around the what? The proteins after they're made by the ribosomes. The nucleus. Now remember, does every cell have every organelle? Most of the time, but do they have the same amount of each organelle? No, because structure is determining what? Function. Smooth ER, very similar to rough, but missing the ribosomes. Its main job is more in terms of like making steroids, like testosterone and estrogen, getting rid of toxins. So we would expect to find a lot of smooth ER in what organ? The liver, maybe the kidneys, mostly the liver. It also helps to regulate certain ion concentrations in your blood, like calcium.
Hey, I have to ask you, what are gonads? Because there was some question about it.
That's like your testes.
Okay.
All right, now what we're looking at here is what's called the secretory pathway, or what else did we call this? The endomembrane system. Good, nice. So we start with a ribosome on top of an ER. It makes the protein. The protein drops down into the ER and gets transported until eventually it's invaginated into a vacuole. It's closed up. So again, like a factory, we're making the stuff. We run it across the assembly line, and eventually we bag it up, and now it's prepared to be shipped out. Typically from there, from the ER, where does it go next? To the Golgi apparatus. All those little vesicles that we just saw in the diagram start to stack up and to make a Golgi apparatus. It's always going to be located where? Near the membrane. Because its job is to do what?
Package it up.
Well, it's already packaged. Its job is to what? Ship it out. And while it's waiting to be shipped, we said to think of the Golgi sort of like a warehouse to some extent. The proteins get modified a little bit. They may get some sugars added to them, some lipids. They get prepared to be shipped out. And that process of shipping out, exocytosis, shipping things out. Now, a key thing to know, and I'll move over. Two kinds of ribosomes in most of your cells. Ribosomes that are on the ER and ribosomes that are not on the ER. The ribosomes that are on the ER are going to make proteins that need to be what? Shipped out. But the ones that are just floating in the cytoplasm make proteins that way. that are going to stay in the cell that made them, okay? That the cell's going to use. All right, there's the Golgi, and again, notice it's right near the membrane. Those vesicles, after they're ready, the proteins are ready to be shipped out, break off, bind to the membrane, out comes the protein. What happens to the vesicle?
Just becomes a part of the cell.
Becomes a part of the cell membrane because it's made of phospholipids. That whole deal from ER all the way to out is called the endomembrane system. And there we see kind of the list of endomembrane system components. The ribosomes really aren't part of it. They're sort of before it starts. Proteins go into the rough ER, are closed up in vesicles, go to the Golgi, come out in vesicles again, and get shipped out. Exocytosis. It's our endomembrane system. Now, lysosomes. You guys know by now that lyse means to what? Break, cut, split, tear, destroy. Lysosomes are containers, vacuoles, that contain really powerful enzymes, hydrolytic enzymes. They got a number of roles in your body. If we're talking about a white blood cell, we talked about this in class this morning. A white blood cell engulfs a germ, a virus, a bacteria. It encloses it in a vacuole. And then what gets turned loose on that germ inside that vacuole? Enzymes from the lysosome. And what do we call the process that's happening to that germ inside that cell?
Intracellular digestion.
Intracellular digestion. Breaking it down, digesting it inside another cell. That's what the lysosomes are in charge of, intracellular digestion. That's important for getting rid of germs. It's important for getting rid of old cells. It's important in things like metamorphosis, when like a tadpole turns into a frog. A caterpillar turns into a butterfly. The way it changes is by breaking down old pieces and then recycling them into new parts. Sometimes the lysosome is called the recycling center of the cell because it breaks down pieces that get reused. Vacuoles, again, we talked about, are storage containers. Most kinds of cells have vacuoles, but plants have this huge one right in the middle, mostly to store water. And what do we call the pressure that builds up inside that? Turgor. And that's important because it pushes out on what? The wall and makes it turgid, stiff, so that it what? So it stands up. Plants wilt when their vacuoles start to run low on water, right? And they become, what's the opposite of turgid?
Flaccid.
Flaccid. They wilt. All right, we talked a ton about the mitochondria, so we're not going to say too much here. We know it's where cell respiration happens, aerobic cell respiration. What two steps in particular? Krebs cycle and electron transport or oxidative phosphorylation. Sometimes it's called the powerhouse. You should know it's got two membranes. The outer one's nice and smooth. The inner one's all twisted and coiled, convoluted. And the inner one is called what? Cristae. And that's where what happens? ETC, electron transport. Remember, the cytochromes are embedded in that inner membrane, and they make up the ETC. And they also make up the pumps for what? What cytochromes pump? Protons, H pluses. Remember, the matrix is the very innermost part of the mitochondria, and that's where what happens? Krebs cycle, also known as citric acid cycle. Also remember that, yeah, pretty much every cell is going to have mitochondria, but some cells, active cells like muscles and sperm, have way more mitochondria than other cells because they need more ATP. Also remember that the mitochondria has its own DNA, shaped like what? A bacterial chromosome, a single circular chromosome. Also remember that it can do binary fission pretty much just like a bacterial cell. It has the two membranes, which might lead you to believe that it used to be a separate cell that got what? Phagocytized. So there's lots of things that sort of tie these into bacteria, and that's one of the roots for what idea? The endosymbiotic hypothesis. That the first cells were what? Prokaryotic, and that bigger prokaryotic cells engulf smaller ones, and over time those smaller ones evolve to become, well, organelles inside a eukaryotic cell. All right, in terms of chloroplast, see if I got a picture up here. I don't have a picture of a chloroplast, but we've talked a lot about them. We know that's where photosynthesis happens. We know that they're filled with little stacks of pancake-shaped structures called thylakoids. And embedded in the membranes of those thylakoids is what? Chlorophyll. That's the sign of the first part of photosynthesis, which is light-dependent reactions, which make three main things. What are they? All right, I heard them all from different people. NADPH and ATP, which are both forms of stored energy. And then oxygen is given off because what got split to provide electrons? Water. Three things. That happens at the thylakoid. Those thylakoids are stacked up. In the stacks, they're called grana. And the fluid-filled space around those is called the stroma. The stroma is where part two of photosynthesis happens, and that's called Calvin's cycle, or the light-independent part. In that part of photosynthesis, basically the NADPH and the ATP, the forms of energy that were made during or converted into the first part of photosynthesis, are used to reduce what? What gets reduced during the Calvin cycle? Carbon dioxide, really, or PGA we might call it. Those get reduced to create what? G3P, which will eventually be used to build sugar, glucose. Also remember that chloroplasts have their own DNA, like bacterial DNA. They can independently reproduce. They look a lot like a bacterial cell. About the same size, about the same shape. Another thing to think about is that cells have a skeleton, a cytoskeleton, mostly made of little pieces of what? A piece of protein. We said sort of like rebar, reinforcement that you might put inside concrete. We might call those little pieces of protein microtubules or microfilaments or intermediate filaments. They're essentially just little pieces of protein. They not only give support, though, they also help to move things around in a cell, like the spindle fibers in mitosis. They grab onto and move what? The chromosomes. It's how the chromosomes get split in mitosis. So important. We're about to talk about mitosis in the main part of class, so we'll hear a lot more about those in a few days. Peroxisomes we didn't talk a whole lot about, but their job is to basically get rid of metabolic waste. Waste products created during cell respiration and digestion. One of those waste products is hydrogen peroxide, which is toxic. So inside these peroxisomes is an enzyme called what? Catalase, which takes hydrogen peroxide and breaks it down into what? Two things.
Oxygen.
Water and oxygen gas. That's the enzyme we used in our enzyme lab. Catalase.
What do you call free radicals?
Don't worry about that right now. We'll get to that later. In terms of cell movement, cells have different ways to move. Some have cilia, which tend to be short and numerous, right? What's maybe the most famous ciliated cell? Like a paramecium, single cell protist. But remember, in people, the fallopian tubes are ciliated so that the egg can be pushed along. The cells that line the respiratory tract are ciliated to move mucus up and down. flagella are a little bit longer than cilia and also usually less numerous. Yeah.
So if those in your respiratory tract start working, is that like why you get sick? Like
Why you get like a smoker's cough? Smoke, cigarettes kills the cells there, so they have to hawk up the mucus instead of pushing it up.
I've been coughing a lot lately, and I started coughing, and I started thinking about
That's probably more just because you have a lot more mucus because of the germs that are making you sick. Nice. Nice. Anyway, in terms of flagella, protists like euglenas have flagella. The only human cells that are flagellated, as far as I know, are sperm. Both of those structures, though, are made of little pieces of protein, microtubules. And they're arranged pretty much the same way in all known eukaryotic cells, something called the 9+2 array. And if we look at a picture here, the reason it's called that is there's two of those little filaments in the middle, surrounded by 9 pairs of filaments. Two in the middle, surrounded by 9 pairs. That's if we cut it and we look down on it like a tree trunk kind of thing. Every eukaryotic cell, their cilia and flagella are made that same way. It's another one of those, what kind of properties? Conserved properties to show us some evolution. All right, extracellular matrix. If you happen to run into that term, we know extra means what? Outside, in this case, the cell. These are materials that a cell makes and then pushes out around themselves. Collagen, which we know is a key protein in what? Connective tissues. It holds you together. It's like the glue that binds your body together. Bone is mostly an extracellular matrix. Materials made by a cell, secreted around the cells. They basically build themselves into a cave. Cartilage is the same way. Not going to say much about cell walls today, but what kind of things have cellulose cell walls? Plants. What about chitinous cell walls? Fungi. What about peptidoglycan cell walls? Bacteria. And the main function of those is to protect against what? Bursting due to too much water. That's the main function. Protect from bursting due to taking in too much water.
how long is this?
Till about 350. I'm not going to talk too much about intercellular junctions today. Inter, though, means what?
In between.
In between. These are connections between adjacent cells. The only one of these that I'm going to mention today is one that we mentioned in class today. And those are communicating junctions. Junctions that allow two cells that are side by side to communicate. In plants, what's the specific name for this kind of junction? Plasmodesmata. And in animals, what's the specific name? Jap junctions. Again, here we're comparing and contrasting plant and animal cells. Up at the top are things they have in common. They're both eukaryotic. They both have a membrane-bound nucleus. They both have most of the same organelles. They both have a phospholipid membrane. Now, there are some differences. Plant cells obviously have that cellulose cell wall that animal cells don't have. Plants have that really big vacuole in the middle. I realize that animal cells have vacuoles. They just tend to be smaller but more numerous. Plants obviously have chloroplasts that can do photosynthesis. Animal cells can't. And something that you'll hear a whole lot more about when we get to mitosis here in a week or two. Animal cells have these organelles called centrioles, which help to anchor the spindle fibers. Plants don't need those, because what do they have that acts as an anchor for their spindle fibers? The wall. So they don't need it. So centrioles, in case of mitosis, sort of take the place of the wall. They're anchor points for these spindle fibers. One other quick thing that I will review you on here. A lot of writing on here, so don't try to read all this for right now. Just listen. Operan's hypothesis. is the idea that at first there was no life on Earth. There were gases, there was water, and there were things like lightning and UV radiation. And supposedly, all three of those things came together to make what? Organic molecules, like what? Like amino acids in the water, the organic soup. And that somehow, over time, those amino acids came together sort of randomly to make what? Proteins. And those somehow came together to make life. And the first life was supposed to be what? Prokaryotic, bacterial, something very similar to bacteria. Now that's Operan's hypothesis. We also talked a lot about this Miller-Urey experiment, which was designed to sort of back up Operan's hypothesis. They took the gases that they thought were in the Earth to begin with, in the beginning, methane, ammonia, water, and hydrogen, ran some electric sparks through those gases, and what they were able to show is that that setup would create amino acids. Now, did they find any life in there? No, they did find some amino acids, but as far as it goes. Now, there's a problem sort of with Operan's hypothesis today, more so with this experiment today. Do they think those are actually the gases that were in the beginning now? Kind of change their mind. So maybe this experiment doesn't mean so much, because they're pretty sure that some of these gases probably were not the ones that were there. Endosymbiosis is something really important for you to know. And it's, first of all, the core idea behind it is what came first. Prokaryotic cells. And that large prokaryotic cells engulfed, phagocytized smaller ones, probably trying to do what to those smaller ones? Eat them. But that somehow those smaller ones got away from being digested and proved to be helpful. And that over time, that symbiotic relationship budded, and they became so dependent on each other that they didn't split up. And that's where eukaryotic cells supposedly came from. Now, in terms of ideas that, evidence that might back that up, there are bacterial symbiotic relationships today. One bacteria living in another. That's provable. Mitochondria and chloroplasts have that bacterial-like single circular chromosome. Mitochondria and chloroplasts can independently reproduce, like binary fission type reproduction. And they both also have ribosomes that look a whole lot like bacterial ribosomes and less like eukaryotic ribosomes.
Has anybody ever tried to take out an organelle and see if it can function on its own as a bacteria?
I'm sure somebody has, but I never have read that. My guess would be no. I mean, because the idea is that this would have happened many, many, many years ago, and that now the dependence is so much that one can't make it without the other. So I doubt they would, but I'm sure somebody's probably tried it, but I've never read an article like that. All right, for our last 10 minutes or so, we are going to flip over and look at cell transport for a minute. And the first thing I want to remind you about, things that can get in and out of the cell membrane very easily are what? Two kinds of things. Small and uncharged, non-polar. Things that have a hard time are big things. What's that probably? Look at the shape. What kind of molecule? Probably a protein. Big things and charged things have a hard time getting in and out of the cell. Either one. So there has to be another way in for those things. Because there are proteins you have to have in your cells. There are ions that you need in your cells. How do they get in? Through channels or carrier proteins. They're either brought in by facilitated diffusion or they're pumped in by, what's that process called? Active transport. Today we'll talk about the two most basic ones, and then we'll leave the heavier stuff for next time. Diffusion. It's the most basic, most important way to move things in a cell. It's movement of something from high to low concentration. It's what kind of process? Passive. Meaning that the cell that it's happening in doesn't have to what? Use its own energy. That doesn't mean it doesn't use any energy. Because if it's moving, it's using energy, right? What energy is it using? What powers it? Thermal energy from where? From the environment. Just heat that happens to be there. Thermal energy from the environment. When we talk about a concentration gradient, we're talking about differences in concentration, right? Diffusion tends to move things down the gradient, which is just another way of saying high to low. Passive. Doesn't require energy, happens on its own, because we know that particles are moving in a random, rapid way. What's that random, rapid movement of particles? Brownian motion. And again, that's caused by what? Heat. Anytime there's heat, molecules and atoms are going to be moving in a random, rapid way. Brownian motion. Let's say you put a cell in an environment and all the things move and it gets to equilibrium. Does diffusion stop once you hit equilibrium? Net diffusion stops, but particles are still what? They're still moving. There's just no more net movement. If one molecule moves in, there's probably one that moves out to replace it, all right? It's random though. Some factors that affect the speed of diffusion. Maybe the most important one is how big the gradient is. The bigger the gradient, what does that mean about diffusion? Faster it happens. The size of the particles. Bigger particles means what? Slower, because they have more what? So I heard somebody say it. Inertia. They're harder to get moving. Temperature. Higher temperatures should mean what?
Faster diffusion.
Faster, because that speeds up what? Particle movement. Let's see. We'll kind of leave off today with osmosis. Osmosis is the diffusion of water. Now we had 4 or maybe 5 definitions for osmosis. First of all, it's a type of diffusion, which means something is moving from what to what? High to low concentration. Water is moving from high water concentration to low water concentration. That's the most basic definition of osmosis. But usually, we don't talk about it in terms of the water. We talk about it in terms of what? Yeah, the solutes that are in the water. So another, maybe more common way of defining osmosis is it's the movement of water from areas of low solute to areas of high solute concentration. If I were to ask you for a definition of osmosis, don't say movement from low to high or high to low. You got to tell me more than that. Low what? In this case, low dissolved stuff to high dissolved stuff. If there's low solutes, that means there's a high concentration of what? Water. And if there's high solute concentrations, there's a low concentration of water. So these 2 definitions say the same thing. They're just 2 different ways of looking at it. Also remember these words over here. Hypertonic simply means more what? More solutes than what you're comparing it to. Hypotonic means less solutes than what you're comparing it to. And isotonic means equal solutes. So yet another way of defining osmosis, movement of water from hypotonic to hypertonic. So that's a 4th way of doing it. And then there's one other way we're going to define it. And next time this is where we'll pick up, because there's some math that we need to look at for this. Let's talk briefly. Actually, let's do this first. First of all, if you put a plant cell in a solution that is hypotonic, what's going to happen in terms of water? Plant cell in hypotonic. Water's going to move in, because water always goes from hypo to hyper. and the cell's going to swell, pressure's going to build up, and the cell's going to become turgid. That's a plant cell. That's the way plant cells normally are, right? Because their roots are normally touching fairly pure water. On the other hand, here we put a plant cell in a hypertonic solution, a salty solution, and it's shriveled up. What's that called? Plasmolysis. Everything shriveled away from what? The wall. Everything shriveled except the wall. That's plasmolysis. That's that word applies to any kind of cell with a wall. It's what happens when they're put in a hypertonic solution, a solution with a lot of solutes. And that's assuming what about the solute? Osmosis happens when the solute can't move, but what can move? Water. Here's the same kind of thing for animal cells. Here we've got an animal cell in an isotonic solution. There's no net diffusion or osmosis. Things are staying the same. Here's an animal cell in a hypertonic solution. If the solute can't move, water does. And in this case, water is going to come out toward the areas of higher solute concentration. The cell's going to shrivel. When there's no wall, that shriveling effect is called crenation. So in plants, it's called plasmolysis. In animals, it's called crenation, the shriveling of the cell. And then finally, in a hypotonic solution, the cell is going to swell up. And if it's an animal cell, there's no protection, so what's going to happen? It's going to burst. Last slide I want to talk about briefly is this one. Water potential. And we'll pick up here next time and do a little math with this. But water potential is important because so far, when we've talked about osmosis, we've only talked about the solute effect, right? What effect do solutes have on water movement? We've sort of ignored this. With what kind of cell is that pretty important? The pressure part. Any kind of cell with what? The wall. That becomes an important variable. So water potential is the idea that you've got pressure inside a cell plus this osmotic or solute potential. Those make up what's called the water potential. What's the symbol for water potential called? Psi. Looks kind of like a trident. And remember, it's made of two pieces. Solute potential plus pressure potential. Now, in terms of osmosis, water always moves from where there's high water potential to low water potential. The water potential is always going to be higher where there's what? Where there's less solutes. Because the more solutes there are inside a container, what happens to that number? The more negative it becomes. And we'll pick up with the math on this next time, but I want to put one more formula up here that I want you to recognize. Slow down. We'll leave when I put this formula up and talk about it for just a second. We also spent some time working with this formula. And I will tell you, this is going to show up on the midterm. And next time, tomorrow, we will look at this formula. That's for solute potential. Equal to negative ionization constant times concentration times R times Kelvin temperature. We'll deal with that tomorrow. Hey, make sure when you exit that you go straight out of the building. Please do not hang around.